Dr. Amit, I'm initiating today's webinar.
Hello.
So,
Oh, I have a nice beach picture behind me. Well, hi, nice to see you.
same there, same there. Okay, so I'll just start. Good evening, participants, and a warm welcome to another series of CIC webinar. And it is a pleasure to welcome Dr. Amit Tiwari from Switzerland. And it's indeed a pleasure. It was a very immediate, very short time we got to know each other. Not at least in the coming couple of hours, we'll know better, I feel. And I have to thank Dr. Saumini for connecting us for this. And for the participants, just to introduce our speaker, Dr. Amit Tiwari is currently working as a medical advisor in ophthalmology department, Novartis, Switzerland. And previously, he was working as an NGO scientist in Sophia Genetics and a group leader in University of Zurich. His postdoctoral fellowship and PhD is from University of Zurich and ETH Switzerland. He has a strong expertise in next generation sequencing and omics technology. His specialties are personalized medicine, in vitro diagnostics, genomics, genetics, clinical and medical information, cancer, ophthalmology, rare disease, biomarkers, scientific lines, laboratory organizations, and people management. Definitely, that's you could say in a very blunt, a hell lot of information that you're going to share it with our participants today. So I warmly want to welcome you for our series. And to our participants, to briefly and for Mr. Dr. Amit to brief about CIC. CIC is Crescent Innovation Incubation Council. We are the armpit of entrepreneurship of Crescent University. So currently we have 61 startups. Out of that, 21 are student startups. And the rest all startups falls under the trust area of life sciences and industry 4.0. So our main focus is mentoring the startups, facilitating them in the investment and grant, identifying the grantees and everything. And the third is helping them in marketplaces nationally and globally. So currently we were able to support about 77 lakhs in grants for our startups and 40 crores. It's a cumulative amount, about 40 crores for raising investment money. So, so this is it. We have started only 1 year back, and in fact, we are still running. And it is a pleasure to have you. And definitely, I'll be catching you up for taking you into a mentor pool also. So, Amit, now the floor is to you. So, welcome. And before just participant, just for your some queries were there. And towards the end of the program, we'll be sending a feedback link. Your attendees and your e-certificates will be dispatched after you fill in the feedback form. And your questions you can raise in the Q&A sessions towards the end. Dr. Amit will be answering to it. Thank you.
Okay, may I start then?
Yes, yes, the floor is yours.
Super. I'm happy to then share my screen to start with. But before I start, maybe a short question. What is the composition of the group like? So how many people here are bachelor's, master's, or PhD students, or are faculty members here, just to understand to whom I'm pitching and to what level of detail I should go?
Yeah, so the majority are from the academic side, and majority is the final year students, I could say, that is postgraduate students. And the second category are the startups who are more related to the medical sectors. And the third is faculties. So faculties are again from the life sector, not just biotech, but medical sciences. And in certain cases, doctors are also there, in practice doctors.
Awesome. So I think my slides would be of interest to everybody because there is a mix from medical to basic biology. So I think we will have something for everyone. Great, thank you very much. Can you see my slides?
Yeah, if you could go in the slide, yeah, perfect.
Yes, super. So thank you very much for inviting me for this talk. It's always a pleasure interacting with scientists, researchers all over the world, and especially at an incubation center, because as you would see in the next slide, I have been involved with startup environment, and it is actually very close to my heart. It has a different energy, a different focus, which I absolutely love. So the title today is Genomics and Precision Medicine. And Molecular Diagnostic Innovation Shaping the Future of Healthcare. This was a brilliant title, actually, I think chosen by either Somini or by you, Dr. Mukund, right? So it was a brilliant. Thank you for giving me this title, actually.
That goes to Somini. We have to, she's the one, the master brainiac.
Yes, so this was a brilliant title. I couldn't have said it better, and I hope my talk does some justice to this title. So yeah, you already gave an introduction of what I have done, so I wouldn't go in details, but what I would like to talk about a little bit here on this slide is my startup association. And so I had started a company called Guru Bazaar, very Indian name, and the idea behind that was to provide a platform for different kinds of trainers, be in sports, education, arts, etc., to have an Airbnb for skill development. So this was something that I had started myself in Zurich here for some time, and it didn't fly off. So then wasn't really the market for it. So this was my first failed startup. Hopefully there will be a few more until I make the successful one. After which I joined Sophia Genetics. Sophia Genetics is currently, was a startup when I joined in. We were 35 people, and currently is touted to be Switzerland's first unicorn. So this was also an amazing environment to work onto an idea, develop it, and share it with the world. And after having done that, I'm now at the real corporate big pharma giant Novartis, in order to also understand how could we bring diagnostics in association with clinical trials, in association with patient therapy and drugs. So going further, patients have always been at the focus of what we do for all of us be it basic science or medicine community. And what I will do is here show you a patient journey of a genetic disease, which helps us understand which are the different leverage points where we could develop ideas, where we could develop solutions that would help these patients. So let's say here patient X. She identifies some disease-associated symptoms. Let's say in case of breast cancer, she identifies a lump or a mass in her breast, or in case of, let's say, Lynch syndrome, which is sort of a colon cancer, where she identifies pain in the stomach, blood in the stool, etc. And what she does is goes to the doctor for a clinical assessment and a family assessment along with the geneticist, the clinical geneticist, to identify whether somebody in her family previously had a certain disease which could have similar symptoms and could be correlated. Having done that, the clinical geneticist and the doctor come to a clinical suspicion of, let's say, breast cancer. And the standard procedure then is that the screening of the breast cancer genes is done, which we call as genetic testing. A report is formed, and then variants could be identified in this report, different kinds of variants which could be likely associated with breast cancer. Now, at this stage, there are two different scenarios possible. Either a confirmed pathogenic mutation is identified, where one knows really that this mutation is responsible, has been shown clinically in the lab, within the clinics, to be associated with the disease, let's say breast cancer here. If that happens, the patient is put into monitoring, the family is tested and monitored, and in case there is a clinical therapy or a follow-up, that is taken care of. On the contrary, it can also happen that no confirmed pathogenic mutation is identified. This happens a lot, actually, depending on the disease. Different diseases have different diagnostic efficiency, so this could range between 40 to 80% for different genetic diseases. So if there is no confirmed pathogenic mutation identified, the patient and the family remain undiagnosed. What that means is we have a set of variants identified in the patient without a clear correlation to a disease. What that leads to basically is going back to the bench, doing research, doing novel mutation identification, then doing the functional verification studies, cohort analysis, etc. And hopefully with all these efforts, one can eventually finally identify a confirmed pathogenic variant in the patient, and then the patient could likely be put onto therapy or a proper monitoring program. So far, so good. Is everybody able to hear me? Is the speed okay? Please raise your hands in case you have any doubt. So far, so good? Can you hear me? Okay. Is it possible?
Yeah, you are audible. You are audible.
Okay, I was asked if it's possible to increase volume at my end. I'll try to be louder.
You can continue your talk. It's okay. We take care from our end. Don't disturb your flow.
Okay, super. Yeah, this is a difficult way to do a webinar where you can't feed off the emotions and the interaction.
I know, exactly.
Yes. Right.
We'll take care of
Cool.
it. Don't worry.
Yes, super. Okay, so having seen the patient journey, you maybe within your startup environment, you can already start identifying where your product fits in, where your idea fits in. If you're a researcher, what you could do. Going further into, or I would say going backwards into genomic sequencing, Sanger sequencing. This has been so far the gold standard, the major power horse of genomics for the last 2 decades or so, 2-3 decades or so. And as you might be already aware, it's a simple reaction where primers and DNA templates are mixed together with DNA polymerases and some nucleotides which have fluorochromes attached to it. And then these nucleotides, they attach one by one to the template, and at each reaction, they emit the fluorochrome which is captured by the detector. After you capture this, basically what one gets is this computational sequence of the fluorochromes, or the fluorographs, and the chromatographs, which actually helps identify which is the sequence in the DNA. Now, a basic technique we have all been doing and was used always in gene diagnostics. And how the process works, in case you are not, you haven't worked in a genetics lab, is you identify a certain gene, let's say a certain sequence, and you amplify that with first with a regular PCR. The maximum you could go again with a very good sort of PCR is, let's say, 800 base pairs, which is already quite a lot. You amplify that, and then you sequence it. But you can imagine a gene X could have n number of different exons. The exons could have quite variable lengths. And so if you have to identify a variant in a certain gene, let's say for BRCA, I think there's about 50 or 51 exons in BRCA1. If you have to sequence each of them for 50 patients, it's quite a lot of work, especially when you're not aware of, let's say, a novel variant, which could be in any one of them. You have to do each of this work one by one. It's quite a diligent work. No, so you have to do this, and imagine, in case you're not even aware of the gene, you have to go through the chromosome or the entire human karyotype to identify a particular variant, a particular mutation in the human genome, which could be responsible for the disease. Now you can see here the limitations of Sanger sequencing, how difficult it would be if you have to PCR amplify each of this thing and then run it through a sequence, with the amount of time and work and resources you require. I'll put it in a very simple way to do some mathematics here. So let's say the total number of protein coding genes in humans is about 20 to 25,000. On an average, are 8.8 exons per gene. So one expects about 230,000 different exons. This is what we call an exome. Now here you can be interactive. Feel free, this is what the fun of this slide is. So if you're, let's say, very good at bench work and you can amplify 10 exons per day, what's the amount of time you would require to sequence all these 230,000 exons? Okay, since I don't hear anyone saying anything, I'll show.
The audio, Amit, we can only reply through the chat.
Okay.
The audio disabled. Yeah.
Okay, right. Okay, so feel free to chat in case you're fast and not an issue. So it's basically 23,000 days, which is 63 years. Let's just say you're very good. It's 100 exons per day. You will still need 6.3 years to sequence 230,000 exons from one person. Let's say you are a superhuman, you do 1,000 exons, it's still less than 1 year. Again, you are a superhuman with supercomputers, super machines, you still need 23 days. Now imagine a situation where a patient with a terrible disease comes, and as a medical doctor, you have to identify the cause, and you have to wait for 23 days before you could actually make a very precise diagnosis and therefore a treatment strategy. 23 days is a lot. Currently, we can sequence the whole exome of about 8 to 12 patients in 3 to 5 days. I'm talking about whole exomes that includes the intronic regions as well. Let that sink in. Imagine this is idea to say how the technology has advanced and which level we can actually support patients today. So I would therefore go back to the patient journey slide, and you can imagine now this entire process with each step has its own time as well. And if the clinical diagnosis becomes a rate limiting step, how difficult it can be for the patient to get treatment effectively. And this step being faster helps patients with genetic diseases massively. Enters what we call massively parallel sequencing in 2007. And what we call, it's the comparison of the cost of genome sequencing has been compared to that of the Moore's law for computing. The Moore's law of computing also stated how computing costs would decrease over time. And this is a slightly older slide until from 2013. However, it holds true still. After 2007, when next generation sequencing entered market, the cost of genome sequencing dropped dramatically compared, let's say, to the first human genome sequence. The cost ran in billions of dollars, with about 15 or 20 different major institutes participating in it. James Watson's DNA was sequenced at 1.5 million. This was in 2008, and already people were very happy that it was only 1.5 million, and there was an article on it. And today, we need about 1,000 francs for the whole genome of one person. So again, this year the slide, as I said, is a bit outdated. 5,000 was in 2013. Today it's 1,000. So again, you can see how technology has supported genetic diagnosis and cost as well for patient healthcare. Right, so there are of course different players in the game. There are different kinds of instruments. There's Illumina, Roche, Life Technologies, etc. Currently, about 95% of the publications, peer-reviewed publications, are based on Illumina machines, so they are the clear market leaders. And what Illumina does is called something called sequencing biosynthesis, and I'll show you in a very simple diagram what massively parallel sequencing does. You take the entire genome, you fragment it through different methods, either by physical shearing or by enzymatic process. You cut the gene into different fragments, and then these DNA fragments are captured on a surface. The surface could be a bead, the surface could be a chip, etc. And here they are amplified to develop what we call as clusters. How this is done is in a simple process. So the DNA fragment is kept, and then the bases T, A, C, G, always in the same sequence, they are fluxed sequentially and about 100 to 200 times. And once that is done, the nucleotides also have a fluorophore attached to it. So after that is done, the nucleotide goes and binds to it, and then an enzyme is flushed into it, which actually cleaves the fluorophore. And this cleaving of the fluorophore leads to a light signal, and this light signal is recorded by the camera here. Now, these light signals are again different colors based on which one can identify which was the complementary nucleotide that went and bound here. So practically the same concept like that in Sanger sequencing, however, done at a massive approach throughout the genome. So once this is done, basically one could identify the sequence, and also the signal strength is directly proportional to the number of nucleotides incorporated. So for example, if you have a stretch of CCCs or GGDs, you would have X times the fluorescence strength. So one could identify that this was a polynucleotide stretch. So what happens is then these images are seen, and these images look like this, whereas this is a snapshot from the top of the fluorescence happening in the image at each point when the bases were flushed in. They are processed, so you can see different shades of it, and then you can exactly know within the clusters where different kinds of nucleotides bound. So you can see the green is for G, T is for thymine, CA blue is for adenine, and guanine was for yellow, and green is for cytosine. You could see these different colors, and then if you focus onto one of them, you can identify at this particular, let's say, cluster, what is the sequence. So you would have basically a C, A, T, C, G, T. At the bottom, it's all C, 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 C. So this helps us identify the sequence massively. Now you can imagine this is done at a massive scale of the entire genome, so requires a lot of computing, a lot of noise is also there, which has to be filtered. So therefore, there is a bioinformatic analysis pipeline that helps us do it. In very simple terms, what the bioinformatic analysis pipeline does, it takes the image, it analyzes it, and then calls the different bases, basically identifying where is the A, where is the G, where is the C, etc. Then these sequences are mapped to the human genome, and there are different tools for that. Some of them are called BWA, BOTI2, etc. And these generate what we call as SAM and BAM files. Then any kind of inconsistency or mistakes are corrected. The sequences are realigned because, of course, we know there are lots of complementary regions, there are lots of similar regions within the genome. So these things are managed at the next step. This is done by tools called GATK or Picard. Once you have that, quality scores are provided to the bases. A quality score, what it tells us whether the sequencing has been done correctly or not, whether that is the right base. So Q score 30 gives a confidence that the error could be 1 in 1,000. This helps us know that the sequence is right, that one could trust this sequence. After this is done, the variant or SNP calling is done. So basically identifying at each position where there's a mismatch from the human genome sequence. This is done by a tool called ANOVAR, and it a tool called GATK, and then the annotation tool is ANOVAR, which actually helps us say whether this misalignment leads to an amino acid change, and what kind of amino acid change, what is the frequency of this variant, etc. Further on, there are different tools which could actually help incorporate expression data, gene ontology information, etc. This helps really make the entire picture complete so that interpretation could be done, which actually leads to, sorry, I think there's one slide probably missing, but this whole thing can be actually done with different kinds of approaches. So it could be a panel-based sequencing. What a panel is, you can choose a subset of genes. So let's say for breast cancer, one knows BRCA1, BRCA2, a few other genes that are involved. So one could choose only those genes and sequence that. This is cheaper, is much more targeted, and has clear advantages in terms of efficiency, in terms of turnaround time, etc. There could be whole genome sequencing where the entire exome is coded, the whole exome sequencing where the entire exome is coded and they are sequenced. This has, of course, much larger size. About the files are about 15 gigs, and there's a potential to identify novel disease-associated variants, novel disease-associated genes, and has, of course, implications for undiagnosed individuals. How this happens, I'll come into later with an example. A whole genome sequencing, of course, where all the coding regions, the UTRs and intronic regions are covered. Of course, this leads to very huge files, very huge data sets, about 120 gigs per sample. And this has, of course, potential to identify variants, large regions that are associated with the disease within also the non-coding region. Of course, the difficulty here is data interpretation and storage. So just to give you a sense of what kind of data output we get. So within my sample of Swiss patients, where we did whole exome sequencing, we had identified 50 to 60,000 variants per patient. And these were distributed into different kinds of variant types. So majority of these variants are missense or synonymous variants, which are single point mutations. Of course, a huge population was also intronic and UTR variants, etc. Quite a small fraction is nonsense and frame shift variants, which are, that's the real strong pathogenic variants often. So this is quite a few number, but majority of the variants lie in the other kinds of mutation types. Of course, there's also splicing mutations that one, splicing variants that one captures. Okay, so now you can imagine we have this huge number of variants, and in retinal dystrophy patients, which are basically monogenic inherited diseases, which means mutation in one gene is likely responsible for the disease. Mutation in one gene, you have to identify from these many different variants. And that is what is the tricky part and what we call as interpretation bottleneck in precision medicine. We have huge amount of data, and we have to identify that one or two variants which are associated with the disease and therefore respond likely to therapy. So the first part of today's large-scale genomic data production, which is processing, filtering, etc., which are all bioinformatics based, are quite advanced, have progressed at a remarkable rate, and one can get enough data quite soon. What is the bottleneck is really the annotation and prediction. So annotation is currently fragmented. There's not enough information and pathogenicity with all different kinds of variants, because of course, annotation and functional prediction are done by basic wet lab reductionist biology, which takes time, which takes effort. And then, of course, that has implications on the interpretation and generation of a final genetic report. So this is what we call as the interpretation bottleneck. And currently, a lot of effort is going towards resolving this bottleneck so that precision medicine could be made available to the larger masses as fast as possible. Of course, this kind of approach has its interpretation challenges, which add to the bottleneck. So I'll take two examples here, one of a monogenic disease, which are inherited retinal dystrophies. Although many of the monogenic diseases are rare, overall they affect a lot of people in the world. What adds to the complexity is that if they are rare, there are very few patients on them, which means there's a lot less phenotypic, genotypic data, symptomatic data out there that one could correlate very quickly with strong statistical significance, with strong associations. In addition, even though they are monogenic, they could also be there could be a lot of heterogeneity. For example, in inherited retinal dystrophies, if you just look at this one corner here, you could see these four genes, ABCA4, PROM1, PRPH2, and RPGR. Mutations in these are associated with either retinitis pigmentosa, or cone rod dystrophies, or macular degeneration. So once you identify a variant, you don't know what kind of disease the patient has, or vice versa. If you know the disease of the patient, you don't know which specific genes to look into. Again, if you were doing this by Sanger sequencing, was a lot more difficult. Today, you can do it a lot faster, but to identify and make precise genetic diagnosis, one has to look into the data a lot in detail. So I do see a question, probably a hand was raised. I'm happy to take it now if there's a question.
Okay, I'll allow Adil to talk. Okay. Mr. Adil? Mr. Adil, you want to raise a question? Adil? I suppose that should have been an accident. Yeah.
Okay. Well, I got a little break to have a gulp of water. Right. So a second example is, of course, cancers, which, as you are aware, as time progresses, often tumors progress and they acquire additional mutations, and that helps them in progression of the tumor. In addition, also upon drug treatment, the tumors, in order to escape the treatment, they acquire additional mutations. So over time, a tumor builds up additional genetic heterogeneity. That becomes very difficult as a geneticist, as a medical geneticist, to identify which has been the driver variants, which the driver mutations, which were the passenger mutations, which kind of treatment combination one could give to the patient based on the molecular profile, the molecular genetic profile of the tumor that one is assessing at that moment. Okay, so this is what I call, once you've done it and you've identified the variant, it's called finding a needle in the haystack. It's difficult, but actually is a lot of fun. Again, coming from personal experience, every time you are able to identify a reason for a patient's disease, not only it's gratifying for ourselves, also for the family that they have now a reason, a cause where they know if they can do something about it, great. If not, they also have an understanding of what could likely happen, and this is remarkable. So to do that, identify this one of few variants associated with disease. I developed a variant filtering pipeline. What it does is it looks at the whole exome data, and let's just say if I'm looking at inherited retinal dystrophies, I know that there are about 250 genes. Now it's actually a lot more number, which are associated with inherited retinal dystrophies. So in this case, I would look for variants in these genes only from the entire collection, and the number, of course, becomes quite a lot lesser compared to the 50,000. Of course, there's also an additional tool which we call Human Gene Mutation Database. This has annotations of known variants with strong significance association with the disease or not. And this is currently owned by QIAGEN, previously used to be available otherwise. And if you look into that, one can identify very clearly very few variants that helps us again narrow down to the cause of the disease. If one doesn't find anything, then of course one can use different kinds of tools. Now, these tools are basically quite logical tools. So let's just say a certain disease has a frequency of being less than 1 in 1,000 in the population, or less than 1 in 5,000 in the population. So one logically looks into those variants that have a frequency of less than that. Then one looks into different kinds of amino acid change prediction scores or protein prediction scores. That some of them are called SIFT, MAP, mutation taster, etc. And what they are basically based on different properties. So whether the structural integrity would change, whether amino acid changes from a basic to an acidic one, and things like that. So then it adds a different kind of a qualitative information and prediction on whether a certain mutation would lead to a strong phenotypic change in the protein behavior. So based on these factors, one could further narrow down the variants. Of course, quality, coverage data, and strand bias are also seen, and therefore the numbers could also be further reduced. So with that, one has a lot less number of variants to work with, and then have a causal association with the disease. Now imagine you do not have specific known genes. If it's a novel gene or there's no disease association so far, one has to do the whole exome analysis, and with all the 50,000 variants, one does a similar kind of steps, and the numbers are quite different. And let's say you end up with something like 6,000 variants still with a certain disease, and this is the tricky part to identify. So of course, one again here uses different kinds of combinations. So for retinal dystrophies, I looked at these genes and I tried to map them to expression data and see whether some of these genes were expressed in the retina. If they were, it made sense that they might have involvement in blindness. Also, gene ontology annotations on functional behavior, etc., were combined together to come to this information. A few years ago, then the American Society for Medical Genetics, American Council for Medical Geneticists and Pathologists came up with what they call the ACMG Guidelines, quite based on the previous slide. I wouldn't go into the details here. What it does is it tabulates the information into different kinds of data input one has to look into. That is population data, predictive data, functional data, segregation data, how a variant segregates in the family, if the variant is de novo, allelic data, etc. And then based on this, helps with different kinds of qualitative information, whether this data is very strong, supporting, moderate, less supporting, etc. And then with that, one could group variants into either benign or pathogenic with different grades of confidence on this benign data or the pathogenic data. You could read up this paper. I'm happy to go into detail in case later also if you have any question. It's quite detailed and quite boring if I have to go through the slides, because I wouldn't go and I would jump into. I'll just show you a brief snapshot of our results. Within the CIS patients that we had, we did whole exome sequencing. As you can see on the left here, we identified different genes with different kinds of disease frequencies and mutations that we identified. Of course, it is dependent on the patient population we had, but it could also have something to do with the genetic background of the patient. So if we had, let's say, the same type of disease subgroups, and we did the same thing in India, we might actually see quite a different distribution. It's likely possible. We had all kinds of disease, all kinds of variant subtypes, so missense variants were the highest. We also had lots of nonsense, frame shift, and splice sites within the confirmed variants in the patients. We had different kinds of diseases in our group, and within all this heterogeneous data, I'm quite happy to say that we had above 65% diagnostic efficiency in inherited retinal dystrophies, which was actually even 80% in case of syndromic inherited retinal dystrophies, where you have, in addition to the main disease, you have syndromic features, which helps you identify and diagnose patients better. We identified about 50% of the variants that we identified, they were novel variants. This would have been very tricky to identify again with Sanger sequencing. So this is something, again, a strength of whole exome sequencing. And we also uncovered lots of novel genotype-phenotype correlations. Again, I would not go into details here, but just to say, with this technology, we are able to, within a very short period of time, get massive data and actually have impact on quite a lot of patients quite rapidly. So it's fast, it's reliable, high success rate, but what else can we get in addition? And here I would take one clinical example, which was very interesting. This was actually my first case that I worked on. This was a female patient, was first examined at 10 years of age and was initially diagnosed with chorioretinal degeneration with high myopia. She's of Iranian ancestry, and her parents were first cousins. The ERG showed that basically she had no cone and rod function in her eyes. So what we did was, because the parents were first cousins, we did a combination of linkage analysis and whole exome sequencing. Having done that, what we did was basically looked into only the homozygous regions in the genome, in the exome, and we identified about 2,327 variants within the homozygous regions, which gave us this sort of a table of different kinds of variant numbers. What was very interesting was then we identified one frame shift deletion. This was a novel one. And a frame shift deletion, as you can imagine, it leads to a shift of the frame and eventually would lead to some sort of a stop codon getting acquired downstream, which often leads to nonsense mediated decay of the RNA. Therefore, the protein would not be produced, would not be formed. So this was kind of the interesting molecule, very, very like in one go, one looks at that and goes like, this could be something to look into. And of course, when we look into it, we identified it was a gene called COL18A1 with this mutation. And mutations in COL18A1 are known to cause Knobloch syndrome. Now, Knobloch syndrome have different kinds of phenotypes. Some of them are actually retinal dystrophy, retinitis pigmentosa, retinal degeneration, high myopia, and vitreochoroidal degeneration. What was interesting was that Knobloch syndrome have patients with what is called an encephalocele. Now, this encephalocele is formed due to a failure of the closure of the neural tube during development. Trigger warning for the next slide in case you're sensitive. This is how Knobloch syndrome patients sometimes end up looking like. This is this mass that develops due to failure of the neural tube closure. So actually, once we saw the mutation in the COL18A1 gene, we went back to the clinician and the family and asked them, did you see that in your child when she was born? And actually, the family had moved from Iran to the US. So they go back into their files, and actually they found that at birth, the kid had that, and this was removed after birth. So this was a very clear example of a variant of a patient who was initially incompletely diagnosed only with chorioretinal degeneration. However, whole exome sequencing helped identify or diagnose the disease correctly. Now, this can have quite strong implications on how the patient would be managed. Just to show you very quickly the data, the patient here had a homozygous deletion, and the parents were both heterozygous. You can see there two different electrograms here in the Sanger sequencing, and this mutation leads to deletion of half of endostatin, which is at the end of col 18A1, and this could also have very interesting consequences on how the patient on the patient's phenotype. Okay, a second example is of a patient with retinitis pigmentosa. This is a 60-year-old female, and the parents were healthy. They did not have any variant, so either the patient could have a de novo mutation, or the parents could both be heterozygous carriers of a certain mutation or two different mutations, but in the same gene. So we performed the whole exome sequencing. However, we could not identify anything within the exons. So what we did was we looked at the padding regions. Now, padding regions are regions just adjacent to the exons, but are part of the intronic regions. And once we looked into that, we identified a variant in intron 5 of the CDHR1 gene, in addition to an exonic variant in the same gene. Now, this was -17 base pairs from the intron 5, so one would normally miss if one would just look at the exon. However, looking at this in detail, we did a segregation analysis, and we could see that the variants were segregating perfectly in the patients. The patient had one variant each from each parent. That made, again, quite interesting sense. So what we did was we did what is called a splicing analysis. Now, what a splicing analysis does is checks whether a certain splice acceptor, splice donor site, is stronger than the canonical one. And on this, you can see this is the reference sequence, and this is the patient with the mutation. And here, there's a strong gain in a splice acceptor site, which is quite comparable to that of the canonical site, a splice acceptor site that you see, which this implicates that there might be splicing happening here, and then this exon would have additional nucleotides. Now, depending on whether this is in frame or out of frame, this might lead to a frame shift deletion or a larger insertion within the gene, which might actually inhibit its function, have effects on its functions in many different ways. So what we did was we inhibited nonsense-mediated decay using cyclohexamide, extracted the total RNA, and then we identified basically in the patient cell line an additional product, which was exactly involving these 15 base pairs from the intron 5 within the patient, clearly confirming that this variant -17 base pairs downstream actually had an impact on the splice acceptor site, and the canonical was canonical splice acceptor site, and it was competing with the canonical splice acceptor site. So with these two cases, what I wanted to show was that with whole exome sequencing, again, the advantage is not just looking into the particular gene, but also one finds additional information which could have strong implications and could actually help diagnosis faster. To summarize, genomic sequencing has, of course, huge implications on precision medicine, diagnostics and research, reproductive decision making, risk assessment of a certain disease, also psychological ramifications. So when often parents are quite stressed when they don't know what has happened to their child, this helps them understand the basis and actually deal with the problem. And of course, pharmacogenomics, and these points I would come into next slides. I would pause here a second and ask if there are any questions until here.
You have captured the crowd, I feel. So far, no questions.
Okay.
Anyone, does anyone need to ask him personally? Can raise their hands. I'll unmute your. I suppose you can continue. Yeah.
Okay, so I either have captured everybody or everybody's sleeping. Okay, nice little CF time. Okay, perfect. I'll go to the next slide. So one of the first examples of a proper precision medicine, as we call today, was the retrospective trial of Olaparib in ovarian cancer patients. Now, this is Olaparib is a PARP inhibitor, which inhibits the PARP pathway. Some of you might already know within our DNA repair system, if there are single strand breaks, the PARP, which is a set of 18 proteins, they come together with the basic excision repair pathway and they correct it. Now, in case this is not functioning, the DNA ends up having double strand breaks or there's a replication fork collapse. Now, if that happens, the DNA has two mechanisms to still try to manage somehow. So one is that the double strand break is repaired with BRCA1, BRCA2, so BRCA genes with a competent homologous recombination mechanism, and the cell survives. Now, if this doesn't happen, let's say if the BRCA genes are mutated, the DNA, the genome has no choice but to use the impaired homologous recombination, which is non-homologous end joining. Now, this mechanism is very error prone. It leads to a lot of different and small insertions, small deletions, small variants in the genome. Now, what that leads to is a genetic instability and therefore cell death. So the PARP inhibitor was actually employed this basic biological pathway, and by inhibiting the PARP, and in case of patients where there was no BRCA due to mutations in BRCA, pushing the cell into going for a non-homologous end joining and therefore genetic instability and cell death, leading to a synthetic lethality of the cell. This is the pathway, and this is what Olaparib does. Now, within patients within the clinical trial for patients who were treated with placebo versus those who were treated with Olaparib, it was identified that the progression-free survival was much higher in patients with Olaparib. So these are patients, it's a maintenance therapy. So after the platinum sensitivity, Olaparib is provided to the patient. So this graph is showing all the patients that were in the trial, and the progression-free survival in the Olaparib patients, as you can see, was 8.4 months compared to that of placebo, 4.8 months. So quite a good survival. Then patients who had BRCA mutations. This was the previous slide was all patients, but in this graph, patients who specifically had BRCA mutations, you can see the patients receiving Olaparib were almost 3 times likely, had a 3 times likely longer months of the progression-free survival compared to placebo patients. And patients that had wild type BRCA, even those there you can see the number is a lot less. However, even there, the patients with Olaparib did have a certain advantage. So one could then identify BRCA mutations in ovarian cancer patients and then preemptively know which patients would benefit from a Olaparib treatment or not. And this is what is the goal and the beauty of precision medicine. So pre-screening these ovarian cancer patients and therefore putting them into a better prescription of maintenance therapies and therefore an outcome. A similar algorithm also exists for non-small cell cancer tumors, which have driver mutations. So NSCLCs with EGFR positive mutations here, depending on the type of mutation that the patient has, one could treat the patients with different types of first-line therapies. If there's deletion in the exon 19 of EGFR, the first-line TKI, tyrosine kinase inhibitor, afatinib is preferred. However, if it's the L858R mutation, there gefitinib and erlotinib could also be given. However, as the disease progresses, often lung cancer patients accumulate T790M mutation. This is a resistance marker for EGFR positive lung cancer, and then first-line therapies do not function in these patients. So therefore, these patients can only benefit with the third generation of EGFR tyrosine kinase inhibitors. Now, as a clinician, as a medical geneticist, if a patient comes, and one could do this in advance, one knows whether a patient already has a T790M or not, one could then accordingly treat the patient. Also, the patient could be followed up during the treatment procedure to make sure that he's given the right treatment at the right time. I do have a question here. The question is, since after genome-wide association study in 2005 for ARMD, how the progress of karyotype-based medicine research and how long it may take it as a common approach in the field for accurate diagnosis for the public, especially in India. So this is coming from Ashish. Hi Ashish, I'm not really sure what is ARMD. Could you please elaborate on that?
Ashish, if you could raise your hand, I'll unmute you. You can directly ask it. Yeah, you can ask. Can you unmute yourself?
Yeah, hello sir, can you audible?
Yes, hi Ashish, you're audible. Yes, I can hear you.
Yes, sir. I'm Ashish. I'm from ICMR National Institute of Technology. Actually, I have come to this karyotype-based medicine a couple of years in an article.
I'm sorry, your voice is Ashish. Your voice is breaking. I'm sorry, your voice is breaking.
Hello.
Your voice is dragging.
Sir, hello.
Yeah, better.
Yeah, it is actually what I meant is that age-related macular degeneration. What I meant is ARMD, age-related macular degeneration, which is the first genome-wide analysis conducted in 2005. Apart from the genome-wide investigations of diseases, how long it may take as a common approach for the some of the diseases which is cancer related as well as what is the field of this karyotype-based diagnosis? How long we may need to wait for a personalized medicine?
So, Ashish, there was quite a bit of a disturbance within the question, but I see your question is for age-related macular degeneration and how genomics and karyotype-based medicine could be helpful there. So, of course, there are a couple of these complementarity factor associations with age-related macular degeneration that were identified with the whole genome association studies. However, that is, if you think of it, that's quite a small part or percentage of all the patients that are affected with age-related macular degeneration. And even though the association is there, there are therapies which are maintenance therapies in a sense that patients have. So I think you're already aware of Lucentis, Avastin, so ranibizumab basically, or there's also from Allergan, that is aflibercept. These drugs are being used and provided to patients, and patients do show quite a good response. So I think it's a very good question of how genomics could support here. What could genomics do? And again, this is again going a little bit in the hypothetical region, because age-related macular degeneration is not my basic research topic, so I'm not 100% sure of what I say. But again, understanding on how it could help with patients if we could identify positive markers within, and then find this association with the treatment approach, and we see, okay, these patients with a certain variant are being able to have better outcome with anti-VEGF therapies. So that could help patients treat them better. So it could have an impact there. Do I answer your question?
Yes, thank you very much.
Super, great.
Yes. Thank you, sir.
Okay, you're welcome. If there's no more question, then I could go forward, but if there is, feel free to shoot. Okay, so I think I could go forward. Perfect. So summing up the non-small cell lung cancer slide again. So basically using these approaches, we will be able to go forward pre-screening patients and identifying variants, and therefore providing them with the right kind of
Amit,
therapy.
you are able to hear me?
Yes, I can hear you.
Hello?
Yes, I can hear you. Can you hear me? Hello. Hello. I'm not sure if you hear me. There is a pause. Dr. Mukund, if you could hear me, could you please let me know if I should go ahead? Okay. Thank you so many for telling me. So I would go ahead in that case. So perfect. Thank you, Ram. Thank you for informing me. Okay.
Hello.
Yes.
So do you hear me?
Yes, I can hear you.
Yeah, I hope all you understand the session. The session was very nice. Then, okay, I will send you the feedback form. Please fill in the short box.
Okay, so if it's fine, I go ahead with the next slide. Okay. So this is the final part that I want to touch, and that is pharmacogenomics, which is also being impacted tremendously with genomics. So within pharmacogenomics, let's say this gene's called CYP2D6. There's a set of enzymes that are actually metabolizers of different kinds of drugs that we have. So depending, and these genes often have different combinations of variants within them. So they can have complete gene deletions, they could have single gene copies, multiple gene copies, etc. And based on these different kinds of mutations, they can acquire different behavior, and the cells would have a different behaviors. For example, if there's a complete gene deletion, the enzyme is not produced, so there's no metabolism of drugs. And there are different kinds of single gene copies where the enzyme could be inactive, normal, or altered substrate specificity, and therefore the enzyme, there would be a decreased metabolism of the parent drug, or normal metabolism, or other metabolites could be produced. And finally, there's a gene duplication. There are multiple copies of the genes. In that case, there would be higher enzyme levels in people, and these people would end up having quite a strong metabolism. They're super metabolizers, and they would basically metabolize any drug very rapidly. So how does this information help us now? This could help us tailor treatments better. So let's just take one example. In colon cancer patients, there are different kinds of patients. Now, currently, or until recently, a one treatment fits all approach was given, same type of therapy, and it would be by rule of thumb, by judging how the patient responds, the dosage would be adjusted. However, now with genomics, if we are able to identify how the therapy would benefit somebody, would they have a stronger effect, no effect, or an adverse effect, one could group these patients into different categories and therefore help them have a more tailored treatment. Now, this is a very difficult gene. This CYP2D6 has quite a different combination of mutations. Again, without going into the details of it, however, these are possible to sequence only through whole exome, whole genome sequencing, where one can go into all different combinations of duplications, gene hybrids, insertions, gene deletions, etc. With that done, one can also identify the different kinds of single synonymous variants, and each of these single synonymous, non-synonymous variants actually adds to the trait of how the CYP2D6 would be metabolically active. So now you can see how it's a very complex, a gene, a very complex system. How do we tailor this to patients? So actually, what is done these days is some very cool things. one could have a pharmacogenomics ID card. Basically, it has a barcode. If I have one, and this is from Holland, from Netherlands, with this information, one can go to a doctor. It has information on the gene status, which kind of, whether they are rapid metabolizers, poor rapid metabolizers, etc. in the patient. And this could be shown to the doctor when going for a doctor visit. The doctor is able to therefore tailor the treatment best for the patient's need. It also has the barcode, which could be scanned on an iPhone, and one could get this entire information of how a patient would respond to a certain therapy. A final topic on how genomics is helping us is genomics in COVID times, and that is why we are having this talk virtually today. As you can see that the Next Train project, just Google for it, and it's quite interesting. It is tracking the SARS-CoV-2 virus and as it travels across the globe. So the project uses genome sequences of the virus derived from different patients, and it generates the map. And the circles, they indicate where the size is of the different viruses. So basically, one could identify a certain isolate, a certain strain of the virus with the strength of the outbreak, the strength of the phenotype over time as we gather more data. Now, this again is one of the largest scale use of genomic sequencing that we are seeing in current times. We are living it at the moment. So with this, I would like to come to the conclusions, and feel free to add in your own conclusions if you have additional ones. So genomics and precision medicine have, of course, implications in diagnostics, implications in personalized medicine, in decision-making on surrounding reproduction, assessment of a certain disease, also identification of novel syndromes or disease-causing genes. So if you have a genome sequencing done now with a certain disease, but you're not able to identify the cause, 10 years later, 1 year later, one identifies through another study association of a variant with the disease. The patient is able to go back to a doctor with this information, or the doctor is able to call back the patient with that information and put the patient on a certain treatment. Which also leads to retrospective clinical diagnosis as well, which is what we did for a lot of our cases. And of course, the psychological ramifications, which I already alluded about. So having said that, the slide would be incomplete if I do not thank my affiliations. So my PhD was at Zurich, and this is the building here, and University of Zurich, where I did my postdoc and I was a group leader at. This is the city of Zurich with a great view of the lake and the mountains. And the University Hospital Zurich, the University Hospital, which provided with the patient samples, the funding, and of course, patients and their families who were supportive throughout this research and research work done. With that, I open the floor for questions. Thank you very much.
That was inspiring. So I have to really congratulate you, because most of our participants are still with us, and we do have a couple of questions. So shall we go directly to Q&A section? I mean, in there, questions have been raised. A couple of questions which you can read
yes.
it out.
True. Yes. So Baby Krishnan asks, how helpful will personalized medicine be in treating and controlling infectious diseases? I hope my slide on COVID might have given you an answer there. So of course, treating might not be immediately available. What one does understand is which kind of variant is strongly associated with a stronger phenotype. That might already help us narrow down. It can also help us when vaccines are being developed, understand if a certain strain responds better to the vaccine or not. So that could be, again, it's purely speculative, but that is the way one could imagine precision medicine going there. A second question also from you, is this concept personalized medicine sophisticated for a country like India, where many of us have minimal access for basic treatment? Very good question. Again, I guess the personalized medicine concept is not a sophisticated concept per se, because one, the costs have decreased a lot for any kind of sequencing. So today, anybody having cancer could, in India, ask for a genomic sequencing. And with that, Of course, you reduce the cost. You do not put the patient and the health system through a tremendous burden of treatment, which is unnecessary, but go for specific treatment. So actually, for a country like India, where there is a dearth of facilities in many centers, there are many centers which are extremely good, but there are centers, there are small towns where there would be problem with facilities. In those cases, actually, it will be an advantage to where patient does not have to go to a doctor 20 times just because the information is not clear. Having said that, your concern has its merit, but I probably am a more half glass full kind of a person there. Dr. Ravindra Sadbhai's question is whether pharmacogenetics mostly depends on enzyme expressed as a gene. So this is one of the components which could be addressed by genomics, and therefore I touched upon that. Of course, pharmacogenomics and pharmacogenetics is a lot more complex field. However, genomics would have its one little say in how it can support it, and it would be quite a crucial say, which one could actually control. The environment factors, etc. one cannot control, but the genetic one could identify and very clearly determine the causes. So it's not exclusive. Does that answer your question, Dr. Sadbhai? Okay. The next is a question from Venil Sumantran. Personalized medicine has led to tailoring cancer drugs for patients, but has it helped any other disease? Very good question. Actually, there are quite a lot of efforts going on for many different diseases. So in case of blindness, for example, there is a new therapy or inherited a gene therapy that has come into where RP65 mutant genes, patients with these genes could be given an adenoviral vector and patients are able to see again. So one has the potential, of course. Now, looking forward, the world looks quite brighter with the introduction of different kinds of gene therapeutic approaches. One could identify specific mutations and then treat them using all the different kinds of gene therapy approaches that we have. So yes, I am hoping for that world there. Do you confirm and the second question from Venil Sumantran is, do you confirm the importance of a variant with knockout studies? Of course, it depends upon the kind of variant it is. So the variant could either be a gain of function variant or a loss of function variant. If it is and one has to deal with the type of variant based on the scenario. Of course, knockout studies are one of the approaches to do that. Last was a comment from Venil Sumantran. Thank you very much. Appreciate it.
Okay, and there are a couple of hands raised. So,
yes.
Ms. Mika, I will just unmute you now. Mika? Okay. Sneha? Ms. Sneha, you want to ask something? You can unmute yourself and you can ask him directly. I suppose we lost most of them. Sajid. So, Mr. Sajid, I'll just allow you to talk.
Hello,
Sajid?
sir. Can you hear me? Can you hear me?
Yes, I can hear you, Sajid.
Okay. How are you, sir? How are you, sir?
I'm very good. Thank you. How are you?
Okay. Welcome. Good, sir. My question is that we are studying only the mutated genes. For example, the COVID-19 infection, there's something differently patients from patients reflections. There are many different observations are there on COVID-19 infection. So like this approachment, we are missing. For example, there are some sudden deaths are there, and we are only approaching the mutated genes to identify what's happening there. Some genes are the normal area without mutation, but they will reflect some problems in the patients. So is there any specific study to find that normal genes, what is happening there? Is there any problems arising from the normal genes for the unexpected reflections from unexpected happenings from the for any infections or any things to be in the future and maybe in sudden death likewise things.
Sajid, there was a bit of a background noise, so it was really difficult for me to reach the crux of the question that you wanted. I would appreciate if you could type it. I would appreciate if you could type it, please, and I will read it and answer you. In the meantime, I could take another question. There was a background noise. I couldn't follow the question.
Okay. Okay.
Thank you.
Okay.
In the meantime, I could take another question if someone has another question.
Sure. Just anyone else you want to raise the question directly to him? Meka, I suppose you have raised your hand again. I'm not able to give an access to her. So please, Megha, can you raise a question in the Q&A session so that there's a technical glitch over here. So just to, meanwhile, he types a question. So we have some student participants also. So now it's a career decision time. When you see over here in the Indian side, now it's the pass out, 12th pass outs are there. They are seeking for their future. So how do you, as a personal, now you are in your advisory on now the coming trend, or now we know we have passed a COVID period, we are still passing through it. So to those kids who want to come into the field, the importance of medicinal, personalized medicine, and what kind of, because now there are many anticipations going on inside the students, whether should I take biotech, should I take medical biotech? So if they want to go into the personalized medicine sector, should they really stick on to bioinformatics? So in general, to the students crowd, if you could be
It's a very good question, and actually this was one question I had in my mind when I developed this patient journey slide. Do you see the slide again? The patient journey slide?
Yes.
Right. So this was actually one of the things that I was thinking. And it's a great question. And depending on really where you're coming from as a background, if you're a basic researcher, there's a huge amount of work in personalized medicine that you could do here, which is in research, identification of novel variants, verification, etc. So this is basically one could do basic reductionist biology and applied biology all their life and have a very strong impact. And often, when we are in the lab, this also happened to me quite a lot. We would get so lost in ourselves and our cultures that we are doing, we are forgetting we are so far from the big picture, which is our work here actually has an impact on a patient sitting somewhere. This is a big area one could get into. One could also get into this part where basically genetics testing. This is a lot of wet lab work, doing the testing, running the samples. This is a diagnostics job. So anybody with a master's, a PhD has that background to go in there. Also with bachelor's, depending on the training that you get, one could get into this field. Of course, the analysis of this part, where I have not gone into detail, the analysis of this part, of course, has work from also bioinformaticians. So one could work in a diagnostic sector as a bioinformatician, as a developer. One could also work in the research sector. One could also become a clinical diagnostician. And I do not know the landscape in India for this. I think in India, one has to have a medical degree or some sort of a additional degree to be able to do clinical diagnostics. In Switzerland, there's something called FMH, a degree that we have to take in addition to be able to write and sign on these reports. Having said that, this is one part of it. Currently, the cool part, in case you're very interested into in bioinformatics and into IT, machine learning has tremendously sped up this entire analysis pathway. So a lot of machine learning algorithms are being developed to do the same job. So you can also go into machine learning approaches, coding approaches, develop tools, develop softwares for the entire process. Does that answer the question? Or
Yes,
you want more specific? Okay, great.
No, it does. So Sajid has raised his question in the chat box.
Yes.
You're able to, should I read it out for you?
I'm trying to open it. I do not see. I am on the Q&A.
In the chat box. In the chat box.
Okay. Sorry for being a little bit of a tech noob here. I will try. Somehow the chat doesn't open here on my side. Could you please read the question?
Is there any genomic analysis for the normal genes to observe the infection reaction on the body, example COVID-19, and sudden death had no answer on the reason for and sudden death had no answer on the reason for the death or the symptoms of the infection. Is there any genomic approach by analyzing the normal gene to identify the genomic changes occurred in the patient, either by following the express gene on that patient?
Right. It's a great question, Seth, and I think it's a great research topic, and I'm sure there's work being carried on this. However, I think it's too early to have lots of results on this. As you can imagine, the entire thing started February, March, and it has been the first few months. Actually, people couldn't even go to wet lab work. So many, at least in academia, many people couldn't. But I think this would be a question that would be followed up. At the moment, unfortunately, I do not have an answer for you.
Okay. Okay, so there is a question in the Q&A section, and maybe we have another 5 minutes. I'll take in some couple of questions more. Sure. So what is a knockout gene? Dr. Avinash has asked.
Okay, so basically you can just basically within a system, you can just make sure that the gene is not there, knock the gene out. So it's just that, and one can do it with different kinds of approaches, basically cutting the gene out and throwing it off, and then seeing that the system does not anymore have a certain gene. So which processes, which functions are getting completely abrogated and not working. So you can associate those functions with the likely knockout of the gene. And one could do this through CRISPR technique. One could do also there's something called silencing a certain gene using siRNAs, which the same effect could be had to make sure to understand the pathway and understand the biology.
Okay. So Dr. Ravindran, I should be clear. What he has, how can we, in the sense, agriculture biotech, I presume, help in pharmacogenetics, genomics?
Wow, that's a good question, and I have absolutely no answer to that. I can imagine a lot of things, but if you're an agriculture biologist, I would love to talk on that. I'm happy to talk on that. I think we can brainstorm a bit.
Okay, so I suppose most of the questions are covered. Now, just for the sake of our participants, now I've asked regarding the students, now coming back to the startups. Now, in your experience, the kind of regulatory constraints that you went through, because nowadays personalized medicine is a new trend. So country to country, continent to continent, everything varies. So the kind of regulatory, because starting from the clinical trial, then coming, you did happen to show 2-3 majors also. So then the psychological factors. So so what are the regulatory constraints, and how do you think, how did you face all those and come out of it? Just a brief about that.
Right. So I mean, I must say the slides the patient data that I showed were from my research work at the University of Zurich. So we of course had the correct certifications to do this kind of work from the health authorities here. But having said that, I would answer the question in a more general way in personalized medicine. As you can imagine, what is your, let's say, core thing that you have in personalized medicine? It's the data of patients. So one can then imagine, of course, that patient data and patient privacy are associated with it, and therefore one has to be very careful how one stores the data, how one uses the data, how data is transferred from one server to another. So these are key questions anybody who is setting up a precision medicine, personalized medicine company as a startup has to make sure that this is taken care of. Again, whatever work this is done in precision medicine area, in genomics area, has a very direct impact on the patient. So one has to also be absolutely certain that the results that is there have been rigorously, so the entire algorithm, entire procedure has been rigorously tested, is under typical CLIA guidelines, under typical health authority guidelines. So all the testing labs are made such that, for example, a pre-PCR lab should not be mixed up with a post-PCR lab, etc. They're very basic things, but extremely, extremely crucial to make sure that for one patient, one is absolutely certain of the genetic background and therefore being able to provide the answers with the information that one has. One cannot also, of course, cannot go into too much of a theoretical discussion with the patient when one is not aware of. And these are decisions best left to the treating physician, to the clinical diagnostics person. They are best trained for this kind of job. So as a startup, one has to also think which area one wants to go into. So if you want to be a tech startup supporting precision medicine, that's a different area. You want to be a diagnostic startup, then that's your focus area. You want to be a patient data collection, storage, assimilation startup, that could be thing. You could be all of them, and then you have to consider each of these different aspects.
Okay, thank you. And we'll come to the last question. And again, KB has raised the question: can CRISPR-Cas technology and personalized medicine use together to treat the diseases?
That's the hope. That's my hope. So I was in my lab. I was doing the same. So we had patients where we had specific mutations, and we could actually ask for skin fibroblasts from the patient, and then see if we are able to do anything. Of course, the technology is developing as we speak, and it's getting better. But one really has the hope in biological science and the power of hardworking scientists in the lab that soon we will be able to have transition of that into medical field.
Okay. I suppose there are two more questions. Can we take that? We have another 5 minutes more. Okay. Yeah, just two more questions. One is Ms. Sudha Lakshmi has raised. Sir, in your perspective, what are the regulatory flexibilities for a startup in European countries in the area of genetic diagnosis that a country like India really needs to look into?
I'll be very honest. I wish I could answer this question. I haven't had the opportunity, the good fortune to work in India because I've been working here. So I don't have a comparison there to see how the setup is in India. The advantage of a setup here, of course, is that there are very clear guidelines. So one could follow those guidelines very clearly and actually run through it for, let's say, a startup system, which actually your organization is an amazing amazing ground for incubation. There are similar startups here where there are experts from legal aspects to funding aspect. They provide guidance and support to startups. So my own startup, which was in the area of Skillshare, you could actually just walk up to one of the venture capitalists and then basically sit down and discuss with him what all we need to know. This kind of ecosystem was actually very beneficial here. And I imagine the same is happening at the IIC. Again, I'm not able to give you a very specific comparative answer because I really do not have work experience in India. I studied there until master's and then I left. So.
Yeah, we do. I mean, regulations are quite tough over here, and going into the personalized medicine is still in the research area. It is yet to go into the mass. Yeah, it's
right.
a tough competition when it comes to a startup. It's a challenge,
right.
not competition. It's itself a challenge.
Right. Okay. Right. Okay. Okay. I'm
So
happy to
I
hear more, actually, learn more from you on this topic, actually.
so I'll take the last question for the day, and it should impress you. What is a gene expression?
What's the gene expression? Okay.
Yeah, Dr. Avinash has taken it.
Okay. Okay. So basically, our genome, our DNA is translated into what we call is the RNA. And this is called, the gene is translated into RNA. This is called gene expression. And this RNA is then converted into a protein, which actually goes and does the job that the body is supposed to do. So this, in very simple terms, this is gene expression. So anything happening into the DNA or our gene has an implication this way, that if there's a mutation in the gene, a proper RNA would not be produced or a proper protein would not be produced. And therefore, the body would not be able to do its job. Again, talking about eye diseases as an example, there's a certain protein in the eye that helps in processing the visual signal. Now, if there's a mutation in that gene, the protein won't be produced, and the enzymatic reaction that is needed to process this visual signal would not happen, and therefore the person would not be able to see. This is what is gene expression, the translation or transcription of a gene to an RNA.
Okay. So you have brought in the dogma. So we'll end up with the the dogma that we always carried from the school days till the down. So again, Dr. Amit, I have to thank you for giving me a wonderful sessions. In fact, message is good, good feedbacks is flowing in, and personally in WhatsApp also. So everyone, and for them to reach out to you, to write to you, can you just your email ID, if it's possible, can you just spell it out, or I'll type it over here?
Yes, it is Amit, my first name, then Tiwari, Tiwari, and then dot ETHZ @gmail.com. I'm also available on LinkedIn, so you can also connect me there. Feel free to do so. I'm happy to connect.
No. Okay. So thank you again. So it's been a wonderful time for me also to know more about it. I mean, we still feel that India is still lagging in this sector. Lots more to go. So hopefully we are going to pull and bring you into our mentor session, so that the startups and the students can have a more interactive sessions in the coming days. So thank you again, Dr. Amit, and again to the participants, thank you for being with us, and I'll end the webinar session now.
Yes, thank you for the opportunity, and thank you for everyone to be there. Almost the end of the day, so everyone, I wish everyone a good evening then.
Thank you.
Bye-bye.